So it's my absolutely great pleasure to introduce our opening speaker today, Eric Wieschaus from Princeton University, who's going to talk about David in Basel, heat shocks and culture shocks.
David and I arrived in Basel about 6 months, separated from Madrid. And this was, David's arrival came at a particularly auspicious time for me. I had started my graduate, is there a pointer? I think I can point. I had started my graduate work in New Haven in the lab of Walter Gehring. Oh. That one? Okay. In the lab of Walter Gehring. When Walter decided to return to Switzerland after 2 years, I had to accompany him, and I moved to Basel with a NSF graduate student fellowship where the dollar amounts quickly fell because of the changing foreign currency, and I couldn't afford an apartment. So for the first 6 months, my first 6 months in Basel, I actually took a job as an au pair, taking care of a family with 3 boys, 3, 5, and 8. And I had 2 afternoons and 2 afternoons and 2 weekends per month, 2 afternoons a week and every other weekend in the month where I would take total responsibility for these boys. And Walter thought that this was potentially taking up too much of my time from the lab, and finally somehow arranged for me to have a Swiss graduate student fellowship. I suddenly had money. David arrived, and the 2 of us needed an apartment. And Erika Wenger, who is Walter's lab manager, found a wonderful apartment for us, and David and I moved in together. I met David. The apartment was in a typical family house on a Rose Street. We had to walk through the family's part of the house to get up into the attic where they had installed 2 bedrooms and in the hallway a table with 2 hot plates. And hopefully I'll have time to reminisce a little bit about that time living with David towards the end of this talk. But what I decided I wanted to do more is talk specifically about David's science that David was carrying out in Basel in the Biozentrum, because science is really what we do, and science is really what David did and always cared deeply about. And this was an extraordinarily successful period in David's career. A number of important papers in important significant journals arose from this work. Exactly these experiments on the molecular biology of heat shock that I'm going to talk to you about today. Now, because this is far enough from my field, even though I'm living with David in the apartment, we talked about these things, but my memory isn't so good, and I have the great fortune of being able to interview one of David's co-workers, Paul Schadl, who worked with him on parts of these projects together. And so Paul is going to give you comments occasionally throughout this talk.
So we arrived in Basel in the spring of 1975. This was our first time in Europe. We went to the lab probably soon after we arrived and met all the members of the lab. There was Yanni Nusslein, David Isch, Hans-Erik Weideli, Spiros Artavanis, and Ruth Stewart. Then there were a couple of technicians, and of course there was Walter, and then the most important person, Erika Wenger, who was the lab manager. Yeah. Walter was trying to transition from being exclusively a geneticist or a rather developmental biologist into a molecular biologist.
So, as Paul mentioned, the major goal and what Walter saw as the advantage of moving from New Haven the Biozentrum was the opportunity to set up a laboratory focused on molecular biology. And these were skills that Walter really didn't have. So he was heavily dependent on attracting very talented postdocs who'd done molecular biology to his lab. This is a 10-foot large-scale painting I did about 3 or 4 months after I arrived in Basel, showing the people who were in Basel. And this is before David arrived, but shows the general structure in the lab, and that Walter... and the general enthusiasm for molecular biology. But of course, actually the real advances happened within the following years when, with recombinant DNA technology, that with Paul Shadle and David Isch and Spiros Artavanis, Ruth Stewart, arrived and actually set up the ability to clone and analyze molecular sequences in the lab. Now, these were early days of molecular biology, and at the time it was exciting and even satisfying to get a gel to work or restriction digest to actually produce the expected fragments of the expected size. And it was very easy for most of the lab to focus on strictly molecular problems. One of the important things, and what struck me about David, was his specific choice, although continuing to actively participate in the molecular part of the lab, to focus on, try to identify a biologically meaningful problem. And the problem that David chose was heat shock and heat shock proteins. Now, the phenomena, the way cells respond to heat shocks had been earlier described by Ritosa. And basically what happens is when an organism like a fly is exposed to unusual temperatures, will shut off transcription and begin the synthesis of a set of about 9 to 10 proteins that are thought to have a protective role in this heat shock response. The most abundant of these proteins was a protein, a 70S protein, and by using RNA collected from heat shocked embryos, previous work had kind of localized that, the gene that was encoding this major 70S protein to 77C. And the interesting feature at the time was one had a gene, a protein, and potentially a single band on a polytene chromosome. So the idea of being able to understand the structural relationships between the genes, proteins, and higher eukaryote was exciting. The other feature, and this was especially important, was that one could actually follow the activation of genes, because this band here, this puff, would dissolve, or this band here would dissolve into apparently an active puff of RNA synthesis. And so what the obvious questions are, how are the genes really organized here? How are they activated? What is the function? What is the significance? And the only real possible approach, certainly much more powerful at the time than molecular biology, was a genetic analysis. And what David set out to do was to analyze, and this is the first genetic analysis of heat shock proteins. And his collaborator in this analysis was Jeanette Holden. And their strategy was first to try to knock out this particular band that was encoded in 87C, and then use that as a first step in developing a genetic analysis. They were able to identify some deficiencies that remove 70, and they had a remarkable property in that embryos, so individuals who are homozygous of these deficiencies, would develop into larvae. And this gave them the opportunity of having an animal that was homozygous for 87C, and they could use that as analysis tools for the heat shock. And so what I remember spending lots of time seeing on a microscope, sorting out and trying to identify these little larvae which were slightly more transparent than the wild type ones crawling around on the plate, and doing heat shocks and molecular biology on this. Paul's comments.
Imagine the disappointment. You collect these larvae for days on end, heat shock them, have your heat shock control larvae, you grind them up, you run a gel, you're expecting that you have deleted the locus that encodes the HSP70 genes, and you're expecting to see the HSP70 protein disappearing. You run the gel, and what do you see when you stain the gel? You see the same proteins that you would see in wild type.
Yeah.
So this, I'm sure, was a nasty shock.
So with that nasty shock, what raised lots of questions. Maybe everything was wrong. Protein didn't map to 87C. And so he and Jeanette then their approach was then to go back, redo the genetics, focusing on 87A. And to do this, they first of all set up an important collaboration with a group in Hungary that were excellent and outstanding geneticists that identified new deletions for both regions. Shockingly, both of them, either of them, when you delete them, had no effect on the synthesis of the heat shock proteins. But then, this is the most remarkable thing for me. Somehow these Hungarians got recombination between these two adjacent deficiencies, made a double deficiency chromosome. When you knocked both out, no more heat shock HSP70 protein. What was eventually shown, and this is largely through the second important collaborator, was that the heat shock genes were largely duplicated, and that actually not only were they present in both these puffs, but there were multiple copies. And from a geneticist standpoint, this actually seriously undermines your hopes of hoping to develop. When genes are repeated and similar in function, you can. And so, and I think I've never talked with David about this, but I think that was in part the push for David to begin thinking about the other projects that you'll hear about through the remainder of this course. He, of course, went on and did incredibly important things with heat shock over the next couple of years. It's largely conserved. Took the Drosophila gene, expressed it in mammalian cells. At this time, heat shock had been also identified in mammalian cells, but under different circumstances. And what was remarkable was that the Drosophila heat shock gene would be turned on in mammalian cells, but be turned on only under the mammalian heat shock conditions. And what that led to the idea was that the cellular factors that turn on heat shock are cell type specific, but the control region of the DNA must be broadly similar in all species that show this heat shock response. It's ultimately led Hugh Pelham's work to identifying the control regions involved in the heat shock gene, and then David used them again in an important set of experiments for developmental significance for looking at heat shock. Now, okay, so that illustrates the quality of that work and the fact that you have these disappointments, or not even disappointments, but exciting new results that make you rethink what's going on, is, I think, characteristic of David's science and characteristic all of his science. And also, I think, to a certain extent, that willingness and openness to new data and new experiences, even though David always brought to those experiences a certain expectation of how things should be and what quality actually, or it characterized not only his science, but also his, in my experience, his living with him. David was extraordinarily tolerant of my own idiosyncrasies during that time. And I'm very grateful to him, but I've come to realize that was in part just natural for David in his approach to life. Talking science was wonderful with David. These are some photos of his conversations with Ruth Stewart, who unfortunately died last year also of cancer. And as you can see, David had opinions, and you didn't always have to accept David's opinions or think they were actually the best. You can kind of see in Ruth's face here. But talking science with David was always wonderful. One of the things that I think about, is that for most of us who've lived our life focused on science and working in the lab and scientific discoveries has been an extraordinarily rich and enriching aspect of our lives. And yet, when I think back on it, in addition to the science itself, what we all come to realize is the wonderful thing that science brings to us, the opportunities to meet and work with other scientists, other humans who are different from us and bring new insights. It's been a great pleasure for me throughout the 40, 50 years since David and I left Basel, or I left Basel to go to Zurich and left the apartment, to meet on a constant and regular basis and catch up, but also get new ideas. So I'll stop there, and thank you all for the opportunity to talk about this.
Very much, Eric. So we do actually have a couple of minutes for questions, if anybody has any burning ones. I'd like to ask one, actually. Your picture of the kind of family that was the Gehring lab at the time is very lovely and heartwarming. But what's the bottle that Walter was holding in his hand? Is it flies, or is it something more alcoholic?
I think when I painted it, I would like to tell you that I had a scientific purpose and it was a fly bottle, and I was trying to imagine, we used to raise flies in glass bottles. It's maybe subconsciously, there could have been a lot, and this is often true for artists, a little, subtle other messages in the painting. But I'm not going to speculate on what those messages should or could be.
Well, I'm sure you can kind of elaborate further on that this evening at the
private
reception. Thank you very much, Eric.